So hi, everybody. Welcome. This is Behind the Scenes of Science, a program for students like you from the Allen Institute. Today, we're going to be talking to Helen Anderson, a research associate in the Allen Institute for Cell Science, which is one of the divisions of the Allen Institute. So just as a reminder, we can't see or hear you, but we will be keeping an eye on questions. So please use the Q&A button down in your Zoom footer to ask questions that Helen will answer later. So the goal of today is that we're going to tell you a little bit about Helen's work and about some careers in science. So we're going to hand it over to her and get started. All right, we're going to go ahead and start with some questions for Helen, and then I see some questions already popping up in the Zoom Q&A here. Please keep those coming. We will have time for those. I'm just going to do a few to get us started. So to start, can you tell us about your science journey from high school to now?
Yes, that is a great question. It wasn't super straightforward. I didn't always know I wanted to be a scientist. So when I was in elementary and middle school, I liked science and math, but I really loved English and I really loved animals. So I kind of thought that I was going to be the next James Herriot. To those of you who don't know who that is, he was an English veterinarian who wrote stories about his sort of work with animals. And I thought that just seemed like the perfect job. So I went to school, high school, and then I went to college. And in college, I declared biology as my major, thinking that I would do pre-med to become a veterinarian. But then the summer after my freshman year, I actually got to shadow a veterinarian, which was really exciting and really interesting. But as I was shadowing, I realized that even though I loved animals, the entire time I was working with them, I couldn't help but wonder about the biology beneath all of the anatomy. I was really wondering, when animal's leg was hurt, I was wondering sort of what the cells were doing. And so when I returned to school, I reached out to some different research labs. And I was very lucky to be accepted into a research lab known as the Rose Lab, which is actually part of the neuroscience department at Western, where I went to college. And at the Rose Lab, I was able to do research with this tiny kind of worm called a C. elegans. And we were studying behavior. So basically, when we present these worms with things they really like and things they really hate at the same time, what do? Are they happy? Are they sad? Are they confused? And I found that I really liked research. I liked the research process. I liked the teamwork. And so after graduating from Western, I stuck with my original major, cell molecular biology, but I didn't do pre-med. And after graduating from Western, I was lucky enough to get an internship working at a cancer research institute. So I knew that I wanted to stick in research. And at this institute, I realized that I really loved cell culture. Cell culture is studying cells artificially in a dish as opposed to in a person or an animal. And I really, really liked that. And so then as my internship was wrapping up and I was looking for jobs, the Allen Institute happened to be looking for a person interested in cell culture, and I was. And it just, I feel really lucky that the timing worked out so well. And I started at the Institute, I guess that would be the summer before last, and I've been there ever since.
Well, that leads us nicely into the next question, which is, what does an average day look like for you, both before the pandemic and now?
Yeah, that's another good question. So I'm pretty lucky in that my work hasn't changed all that much, just the way that we do it. So as I mentioned, I work with cells. So I'm one of the scientists that actually puts on a white coat and sleeves and gloves and heads into lab and, deals with pipettes and test tubes and whatnot. And the cells that we work with are alive. So for those of you who have dogs and cats and fish and guinea pigs and all kinds of pets, you know that whether you're on a school break or going on vacation or staying at home, your animals need to be fed and cared for and watered and taken for walks and given little pets where they get very whiny. Sushi does. And cells are the same way. So whether we're working from home or not, they still need to be fed and they still need to have their needs taken care of. And so before we all started working from home, we would all go into lab together, my coworkers and I, and we would feed the cells and take care of them all together. We would collect the data all together and we would discuss the data all together, all in lab. Now what we do is we kind of have shifts. So someone will go in on Monday and take care of the cells and collect the data, and someone will go in on Tuesday and collect the cells and take care of data and everything like that. And then we all share it online and discuss it from home. So sort of in the same way that you went to school before this and now you're going to school still, it's just different. It's kind of the same with me for work.
What is your favorite thing about your job?
That's a third great question. There are a lot of things I love about my job. In terms of scientific phenomena, one of my favorite things is that as part of the stem cells and gene editing team, one of the things we do is we take those special cells I talked about, the human-induced pluripotent stem cells, and we turn them into another kind of cell known as a cardiomyocyte or a heart cell. And in the same way that you know that your heart beats to pump blood, you can feel it beating in your chest. You can actually see heart cells beating in a dish if you look at them under a microscope. And that just gives me a little thrill every time that you look under the scope and you can see the cells sort of beating in this tissue. It's really exciting. So that's probably my favorite scientific phenomena. But my absolute favorite thing about my job is the friendships, the teamwork, the coworkers. I just really love the people that I get to work with.
Well, science is wonderful, but sometimes it doesn't always go according to plan. What has been your best science failure? Something surprising, a great learning experience, funniest, something like that.
Yeah. So I have kind of two that come to mind. The first is just a funny one, and the second one was a really important moment. So the first little funny science story comes from the lab. So when I was in college, I was helping a friend with a research project where he was using flies as his model organism, a kind of tiny little fruit fly known as the Drosophila fly. And they were very important flies because we were studying their behavior, and they were, he was breeding them specifically. And one day he just wasn't thinking and accidentally opened the vial with all of the flies. And so it got out, and this is what we're studying. This is a very important fly. So we're like leaping around the classroom trying to catch this fly. We did catch him without squishing him, but that was kind of a funny moment where you don't really think about scientists running around the room trying to catch a fly. But that was kind of a funny moment. And then another moment that was really important for me that happened actually in the classroom and not in the lab was in college. So I was taking a biochemistry course, which is one of my favorite subjects, but was really, really hard for me at first. And my grades were always important to me in school. I was one of those people who probably cared about them a little too much, And I was taking this class and I failed a quiz. Like I got an F on a quiz. And I had never failed a quiz before. So I went to my professor's office and I was like, hi, should I change majors? Should I drop out? Am I stupid? Like, what is this? And it was really important because he was like, no, you just failed a quiz. People fail sometimes, and that's okay. You use it as a learning opportunity. You grow from it. You maybe get a little bit of work-life balance in your life, but you definitely should know that it's okay to fail sometimes. You just get back up and keep trying. So that was a really important one for me too.
That's a great story. So now we have some questions from the audience. We're going to get through as many of these as we can, so please do keep them coming. So our first one is from Mr. Scopin. I'm curious where the hipscs come from. Are they human cells? Let's start with the first part of the question there.
Yeah, that's a great question. So hipsc stands for human-induced pluripotent stem cell. So if we break that down, that H for human means that they come from a human. The I, induced, has to do with the process by which we create these hipscs. So essentially, we take skin cells from a man, and then we take them back in time, and we turn them back into something known as a pluripotent stem cell. So pluripotent means that that cell can then become a lot of different kinds of cell. So once we take it back in time, we can turn it back into a skin cell if we want, or as I mentioned that we do in our lab, we can turn them into heart cells or liver cells or kidney cells or any kind of cell you want to study. And then the stem cell part also has to do with the fact that it is pluripotent and able to differentiate, and it also can kind of duplicate itself nicely. So yeah.
Yeah. So and then the second part of the question is, how do we manage the ethics of research? So just very briefly on that.
Yeah, no, that's a really, really important question. And I think it's one that we should take very seriously as scientists. There are a lot of really good books that talk about this. One that I'll plug is The Immortal Life of Henrietta Lacks. I believe that's by Rebecca Skloot. Yes, okay, that's a fantastic book, and it really discusses the ethics of science as well. That's one of the reasons that we like to use hipscs. So because they're human-induced pluripotent stem cells, we get them from a man who willingly donated his skin cells. So that's a really important thing for us, is making sure that there's consent given for all of the resources that we're using, whether they're human tissue or otherwise. Also, I think open dialogue is a really important part of the ethics of science, being very honest and very transparent about what you're doing and getting input from a lot of different people. So I think that for science to be really ethical, we need to make sure that people other than scientists are weighing in on what we're doing. And I think the Allen Institute really tries to make sure that the community is involved in the work that we're doing.
It's a great answer. Thank you. We have another question about the cells. So how do you tag the cells when adding the fluorescent dye? So I think if you can clarify the difference between the tagging and the dye, that would be great.
Absolutely. That's a fantastic question. Yes. So there are a lot of different ways you can highlight structures within cells. And the first that I mentioned, tagging, is a gene editing process. So we use a special pair of molecular scissors known as CRISPR-Cas9. And we go into the cell with these fancy molecular scissors and we tell them where in the genome or where in the DNA we want them to edit. So what we do is we take these scissors and we say, okay, I want you to go into the DNA and I want you to cut right before this certain protein, which is a part of this important organelle, which is a part of this important process we want to study. And then to that protein, we want you to attach a fluorescent protein, which will then light up later when we image it. And by doing that, we make sure that that protein, which then becomes part of that structure, is easy to see. And then when we're studying the process where that structure is important, we can therefore observe that structure throughout the process. So that's gene editing. And that's one way to make a structure easier to identify. But you can also do something called staining or adding dyes. And that's a way that you can take the cell, and this is not gene editing, but you can use chemistry to then affix a dye to a certain kind of protein or structure, making it easier to see again when you image it.
Great. We have another question about the fluorescent dyes. Does the fluorescent dye contaminate the cells, and does it alter the cells in any way?
That's another really important question, and one that we try to be very careful of, because you want to make sure that you're studying cells as close to their natural environment as they can be. Of course, we're working in a lab, so we're aware that the cells are going to be different than they would be in a person, but you still want them to be relevant. And so it's really important to consider the effects that your tampering is having on the cell itself. And yes, the things that we do to our cells can influence them, but we try to be very careful to make sure that they're not actually changing the biology of the cells in a way that is important or significant to the processes that we're studying.
How does your work at the Allen Institute stand out to you as a job?
Oh, as in, like, what do I find most interesting about it? Or
I think maybe what do you find most interesting, or what do you find special about it? Something like that.
okay, yeah, that's a great question. I think one of the things that makes working at the Allen Institute so special is that at the jobs that I've previously worked at in my, lab at college or working at the Cancer Institute, it was very specific. At the Cancer Institute, for example, I was looking at specific chemical combinations for treating cancer cells. And in the Rose Lab, we were studying, very specific sort of behaviors. I think what's really interesting about working at the Allen Institute is that the Allen Institute is dedicated to basic science. So basic science is just sort of how cells work. It's not anything super, super niche or specific. We're not trying to cure one specific disease. We're just trying to understand more about how cells function. And I think that having such a beautiful, broad goal is really interesting and really important, and a privilege that I really appreciate.
Thank you. Another question about the cells we use. Why do we use hipscs rather than another type of cell?
Yeah, that's a fantastic question. These are all great questions, and I hope I'm answering them all right. But we use hipscs because they are pluripotent, meaning that they're able to become a lot of other kinds of cells. So that way we can edit the hipscs, add our fluorescent tags, tag those structures, and then we can differentiate or transform them into cells of different types and then study them in that context as well. So we can study them as stem cells, then we can turn them into other cells, heart cells, for example, and then study them as heart cells in that context. So there's a lot of possibility when you're using a cell that can become other kinds of cells. So that's one of the main reasons that we use them.
We have a related question to that. What is the Institute's main goal in studying these cells?
That is another great, tricky question. because I mentioned that the Allen Institute studies basic science, we don't necessarily have, one overarching goal, like we want to cure a certain disease or we want to research a certain process. But the main reason that we are studying these cells is that it's important to understand what healthy cells are like so that we can help other scientists study things that are more specific. So the Allen Institute has a lot of different outputs, things that we produce, things that we put into the world. Some of those are publications about our research. So for example, we studied how cells are different from one another in a normal, healthy person. And that's important because then we can tell sort of what variation is healthy and normal and what variation begins to enter that sort of like disease illness territory. So that's interesting, putting out those publications that then allowed to us to like contribute to the scientific dialogue. We also put out those tagged lines. So once we tag a structure in a cell, we are dedicated to making those open to the public, open to other scientists to work with, so that everyone can benefit from these structures that are easier to see and easier to study. And then we also put out tools. So our microscopy team and our imaging team and our computer science team, they have these amazing tools for data analysis that we also share with colleges and universities and other scientists, really making sure that we keep that scientific dialogue going. I hope that was helpful.
We have several questions along the same lines that I'm going to group together for the next one. About how does CRISPR treat cells for like gene therapy. So the Allen Institute does not do research on gene therapy. So maybe you can speak more about how we use CRISPR relative to the gene therapy use. But can you speak a little bit to how CRISPR does that?
Yeah, no, that's a great question. Gene therapy is really interesting. So for those who haven't heard the term before, gene therapy is the idea that we can use gene editing and specifically gene editing technologies like CRISPR-Cas9 technology to cure human diseases. So in the same way that oftentimes chemicals are used in processes like chemotherapy for treating cancer, some people are wondering if we can just use CRISPR-Cas9 through a process called gene therapy to go into cells and sort of cut out whatever is making people sick and make them healthy in that way. So gene therapy is a very interesting, very promising process. One of the things that makes it different from the way that we use CRISPR-Cas9 is that we use CRISPR-Cas9 to edit human-induced pluripotent stem cells to make them easier to study by tagging structures and organelles that we then are able to more easily identify under a microscope. But our cells are never intended to go into another person again. So when we edit these cells, we use them strictly for research processes. So they're not actually used in direct medical application. They're used for medical research, but you wouldn't actually see someone ever putting these cells into a person as part of a treatment. CRISPR-Cas9 in gene therapy is actually designed where the gene editing isn't occurring in a cell in a dish, but in gene therapy, that CRISPR-Cas9 is actually going into cells in a person as part of a treatment plan. So again, I hope that was the answer you were looking for. Both are very interesting, very important applications of this technology.
Yeah, and basic research really helps support the development of treatments that are used in humans to treat different conditions. It all starts with basic research that helps us understand how cells work.
Absolutely, yes. And we can even use CRISPR-Cas9 in gene editing the way that we do at the Allen Institute, not just to tag structures, but to create cells that mimic diseased cells. And then other scientists can use these disease cell models to study the potentials of different drug therapies or gene therapies in a dish before trying them out in a person.
Yeah. We have another couple of related questions here. What's been the hardest part of becoming a scientist for you? Is there a specific moment where you've been told to stop working towards where you are now that affected you? And related, have you run into any sexist stereotypes about being a scientist?
Yeah, these are all really, really great questions. And definitely there's truth to all of them. Yes. So what was the first one again?
The first one was what has been the hardest part of the path? Yeah.
Yes. I think for me, the hardest part of the path kind of has to do with the other two parts of that question, which is self-doubt and allowing doubt to kind of creep in. So yeah, I've always liked science, but I've always really loved English as well. English as a hobby, as a subject that I really enjoy. I love scientific literature. I love non-scientific literature. And one of the things that I think was very kindly meant, but kind of tricky sometimes, was how often I was encouraged to pursue that by professors, who were saying like, oh, I feel like maybe you should do this as a career. And then it's kindly meant and it's very supportive and encouraging, but you begin to sort of doubt, like, maybe I shouldn't do science. Like, is the implication that maybe I'm not good enough for science? Am I not smart enough? And so I think the self-doubt is hard. There were definitely times that I encountered sexism or other kinds of difficulty based on, personal factors that are beyond my control, where people thought for some reason or another I wouldn't fit the mold of a scientist. And again, I think one of the things that was really helpful for me in overcoming those many, many moments was kind of not listening to the voices in your head that doubt you, and instead listening to the voices in your life and the voices inside of you that are supporting you, that are telling you you can do it, that you should stick with your gut, you should trust your instincts, and that you are smart enough and that you are good enough. And imposter syndrome is real. For those of you who haven't heard of imposter syndrome is commonly experienced by people who are in fields in which they are not necessarily a dominant presence. And it's when you feel like you are not good enough for that field, even though there's no proof or facts that actually support that feeling. But nevertheless, it is a feeling, and feelings can really influence the way that you succeed. And so I've been very lucky that I've had a lot of support professionally and personally from friends and family, as well as professors, and at the Allen Institute too. So yeah, I feel like that was kind of a long and winding answer to say that all of that doubt is going to happen. It's natural. It's tricky. It's hard. It's frustrating. But you should just, you should trust your instincts, believe in yourself, and also trust the people that are telling you you are good enough, because they wouldn't say it if it wasn't true.
That is a fantastic answer. Thank you. We have a couple of questions now that are back in cell biology as opposed to careers. Before the process of mitosis during interphase, where are the centrosomes?
Yes, this is a great question. Wow. I mean, I'm not a centrosome specialist, but I believe that the centrosomes are just, I think that I'd always just assume that they're hanging out in the cytosol, which is sort of the cell body, this center area of the cell, not the nucleus, sort of the surrounding. I think the centrosomes, I'm not quite sure, actually. I'm not a centrosome specialist, but I should look that up.
Yeah, that's a great question.
Great question.
Another question, how do you direct a fluorescent tag to a specific organelle? Does somebody do it manually, or do you figure it out with a computer?
That is a great question. And we use computer software, which is really helpful to help us design the probes. No, we're not the probes really, but we use a special software that helps us design the instructions that direct the CRISPR-Cas9 scissors to the right place in the genome to add the tag, to insert it. And we do use a computer software program that helps us design that and make sure that the directions that we're giving our cell in the form of RNA are accurate and are able to correctly target that fluorescent protein to the protein of interest, and making sure that that protein of interest will indeed become part of the structure of interest.
We have another one about what is happening in parts of the cell during mitosis. So we had, what are centrosomes doing during interphase? Now the opposite side, what are all of the other organelles during mitosis?
That is another great question. Yes. So they're all still doing their thing, because even though the cell is dividing, it also still needs to keep itself alive. So you're still creating proteins that are involved in other cellular processes that are not mitosis. So, your ribosomes are still doing their ribosome thing, and your Golgi apparatus is still functioning. And the nuclear membrane is breaking down, of course, and then getting rebuilt. So there's a lot of change happening in some organelles, but other organelles, not necessarily a lot is changing for them during this process, but they're definitely still keeping themselves functioning, keeping themselves active, and keeping the cell healthy as it divides.
So we have a lot of questions left and not very much time remaining. So we'll get through a couple more, but you have Helen's email if you would like to reach out and ask more questions. So when did you decide that you wanted to study science further, and how did you decide what type of science to study? You covered that a little bit, but maybe a little more about your thought process.
Yeah. So I would say that I think you guys are in eighth grade, I believe. And it was my eighth grade science teacher that really influenced the decisions that I would later make in college and beyond. She was an absolutely amazing teacher. She was so passionate and she was so positive about all of her students and all of our potential. And I just remember really loving her biology class. So when I was still thinking about veterinary medicine, I was thinking, what are some good STEM majors? There's physics, there's chemistry, there's biology. All of those are important. Math is good. So I was kind of like, what do I want to do? And when I was picking between all of those sort of medical related fields, I was thinking biology made sense in terms of that application. But I also just remembered really enjoying it in middle school. And I think that it's important when you're picking a major or picking a field to study that, yes, you think it's important in terms of its impact on the world or your life or career opportunities. But I do think it's important to also consider what you like. And I really liked biology because this teacher taught me to love it. So that was one of the reasons I picked biology over some of the other sciences, which are all fantastic.
Well, on that note, if you were to choose another type of science to study, what would it be?
I've always had a soft spot for physics. I really love physics. I had some amazing physics professors in college that were just really interesting and really passionate. So I would say physics, definitely.
All right. Well, our last question will be, what do you wish you had known when you were in eighth grade high school about a career in science that you'd like to pass on?
Yeah. I think one of the things that I learned that I realize now is really important, but I didn't necessarily know then, is that experience outside of school is really, really helpful. So if you can, when you're at college or during your summers, look for internships, look for shadowing opportunities, talk to your professors, talk to research labs, really do what you can to get experience outside of the classroom in science. Maybe that's volunteering at the hospital, maybe that's volunteering in a research lab. Maybe that's just shadowing a doctor or a scientist, talking to teachers, but definitely do what you can to learn more about what it's like to be a scientist outside of taking science classes in the classroom, because that's also really important.
That's a great point. Learning about science from a book is very different from working as a scientist.
Yes. And also working as a scientist is very different than working as an academic science student. It's one thing to learn about mitosis in a book and memorize all of the steps. And it's another thing to study cells as they're undergoing the process of mitosis. And I think I didn't quite realize how different those were going to be. Both very important, but very different.
Yeah. Giving that, learning what you learn in class gives you the foundation to become a successful researcher and to
Absolutely.
push the boundaries of what science knows. Yeah, you're studying things that aren't in a book yet.
Yeah, it's scary and it's new. And as you heard with my failure to completely and honestly answer the centrosome question, sometimes you forget the things that you used to know in the book, and you can always go back and look them up again. That's what I'll do right after this. But that's also important to know.
Do you have any final thoughts that you'd like to share with the class?
I don't think so. Thank you again for having me, and it was lovely to chat with all of you. I guess the only thing I would say is keep doing what you're doing, working hard, studying, staying curious. These are unusual times, but remain optimistic for the future because you all have very bright futures ahead of you, whether in science or in a completely different field.